Gluconeogenesis, formation of new sugar. 1. Why gluconeogenesis? 2. How different precursors enter gluconeogenesis. 3. The synthesis of glycogen, starch and sucrose, also lactose. 4. The role of sugar nucleotide. Gluconeogenesis. Gluconeogenesis happens in all animals, plants, and fungi. All the reactions are the same except the regulation. In higher animals, gluconeogenesis happens in liver and renal cortex. Gluconeogenesis can also happen in brain, skeletal and heart muscle. However, liver and kidney remain the main site for this pathway. In liver, the major function of gluconeogenesis is to maintain blood glucose. The gluconeogenesis described here is the mammalian pathway. Why gluconeogenesis? Brain, nervous system, erythrocytes, testes, renal medulla, and embryonic tissues can only utilize glucose from blood as their major or only energy source. Between meals and during longer fasts, or after vigorous exercise, glycogen is depleted. In order to keep the above systems functional, organisms need a method for synthesizing glucose from non-carbohydrate precursors. Non-carbohydrate precursors for a gluconeogenesis. Animals. Plants. Lactate. Glycerol. Pyruvate. Glucogenic amino acids. Stored fats. Stored proteins. Microorganisms. Acetate, lactate, propionate. 3 reactions are irreversible in glycolysis and must be bypassed during gluconeogenesis. Glucose 6-phosphate, hexokinase. Glucose 6-phosphate fructose 6-phosphate, phosphorhexosisomerase. Fructose 6-phosphate fructose 1,6-bisphosphate, PFK1. Fructose 1,6-bisphosphate dihydroxyacetone phosphate plus glyceraldehyde 3-phosphate, aldolase. Dihydroxyacetone phosphate glyceraldehyde 3-phosphate, triose phosphate isomerase. Glyceraldehyde 3-phosphate 1,3-bisphosphoglycerate, glyceraldehyde 3-phosphate dehydrogenase. 1,3-bisphosphoglycerate 3-phosphoglycerate, phosphoglycerate kinase. 3-phosphoglycerate 2-phosphoglycerate, phosphoglycerate mutase. 2-phosphoglycerate phosphenol pyruvate, enolase. Phosphenol pyruvate pyruvate, pyruvate kinase. These irreversible reactions will be bypassed in gluconeogenesis. Reactions in gluconeogenesis. Pyruvate phosphenol pyruvate. Phosphenol pyruvate 2-phosphoglycerate, enolase. 2-phosphoglycerate 3-phosphoglycerate, phosphoglycerate mutase. 3-phosphoglycerate 1,3-bisphosphoglycerate, phosphoglycerate kinase. 1,3-bisphosphoglycerate glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dihydroxyacetone phosphate, triose phosphate isomerase. Dihydroxyacetone phosphate plus glyceraldehyde 3-phosphate fructose 1,6-bisphosphate, aldolase. Fructose 1,6-bisphosphate fructose 6-phosphate, fructose 1,6-bisphosphatase. Fructose 6-phosphate glucose 6-phosphate, phosphorhexosisomerase. Glucose 6-phosphate glucose, glucose 6-phosphatase. 3 bypasses in gluconeogenesis. Because both glycolysis and gluconeogenesis happen in cytosol, reciprocal and coordinated regulation is necessary. First bypass, from pyruvate to phosphenol pyruvate, PEP. There are 2 pathways from pyruvate to PEP. The major pathway uses pyruvate/alanine as glucogenic precursor, however the second pathway will dominate when lactate is the glucogenic precursor. This step involved both cytosolic and mitochondrial enzymes. Main pathway of the first bypass. 1. Mito. The carboxylation of pyruvate by pyruvate carboxylase activates it, initiating the process of gluconeogenesis. Pyruvate carboxylase uses biotin as a carrier of activated HCO3. Reaction mechanism of pyruvate carboxylase. Structure of pyruvate carboxylase. Reaction mechanism of pyruvate carboxylase. Reaction of pyruvate carboxylase happens in two phases, which occur at two different sites in the enzyme. Biotin is covalently linked to the E-amino group of a Lys residue and acts as a flexible arm between two active sites. Main pathway of the first bypass, 2. Oxaloacetate, OAA, produced by pyruvate carboxylase is then transported out of mitochondria in the form of malate. Mitochondrial membrane has no OA transporter. OA plus NAD plus H plus L malate plus NAD plus Malate dehydrogenase. Main pathway of the first bypass, 3. After OA left mitochondria in the form of malate, it will be converted back to OA by the cytosolic malate dehydrogenase, P.546, equation 14-6. This reaction also brings NAD from mitochondria to cytosol, which will help gluconeogenesis to proceed in the latter stage. Main pathway of the first bypass, 4. OA is then converted to PEP by PEP carboxygenase with GTP as the phosphoryl group donor. The same CO2 that activates pyruvate at the first step is lost during this reaction. Alternative pathway of first bypass, when lactate is precursor. Lactate produced from erythrocytes or anaerobic muscle will be converted to pyruvate first by lactate dehydrogenase, LDH, in hepatocytes, the reverse of lactate fermentation. Alternative pathway of first bypass, 2. Pyruvate is then transported into mitochondria, where it is converted to OA by pyruvate carboxylase. Alternative pathway of first bypass, 3. OA is then converted to PEP by mitochondrial PEP carboxygenase, encoded by separate nuclear gene. Second bypass, conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. Because the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate is highly exergonic, the reverse reaction in gluconeogenesis is catalyzed by a different enzyme, FBPAS-A1. Third bypass, conversion of glucose 6-phosphate to glucose. Similar condition also happened in third bypass. Dephosphorylation of glucose 6-phosphate yielding glucose is catalyzed by glucose 6-phosphatase. However, this reaction does not happen in every tissue. Third bypass. Glucose 6-phosphatase is found on the luminal side of the ER of hepatocytes and renal cells. It is activated by Mg2+. Muscle and brain tissue do not contain this enzyme and so cannot carry out gluconeogenesis. Gluconeogenesis is energetically expensive, but essential. For glycolysis, every glucose generate 2 ATP and 2 NAD, P.548. However, 6 ATP, 4 ATP plus 2 GTP, and 2 NAD were spent to generate 1 glucose from 2 pyruvate, P.548, equation 14-9. The extra energy spent is to ensure the irreversibility of gluconeogenesis. Many amino acids are glucogenic. Pyruvate alanine cysteine glycine serine tryptophan asparagine. Aspartate. Phenylalanine. Tyrosine. Glutamine. Arginine. Glutamate. Histidine. Proline. Isoleucine. Methionine. Threonine. Valine.